Mr. Wartsky here again, and welcome to the third screencast of the Organic Molecule series. This is the last slide that you should have seen in the second screencast, so let's go on with the third organic molecule, which are nucleic acids. Now, nucleic acids are the largest of all the organic molecules, and you know them as DNA and RNA. DNA stands for deoxyribonucleic acid, RNA stands for ribonucleic acid. And those should sound familiar from biology. Now, the basic unit of the monomer of RNA is called a nucleotide, and the monomer of DNA is called a deoxynucleotide. And we'll talk about the difference in those in a moment. But they are both made up of subunits, three subunits to be in fact. The first is a pentose sugar. Now, the pentose sugar is what is different in the ribose and deoxyribose. Pentose sugar has 5 sides. Ribose has an extra oxygen, and deoxyribose has one less oxygen. That's why it's called deoxy. There's a phosphate group, and then nitrogen bases. We have either the double-ringed nitrogen bases, which are purines, which are adenine and guanine. And then you have the single-ring nitrogen bases, which are pyrimidines, cytosine, thymine, and if you're talking about RNA, then you need to discuss uracil. Now remember, you can pause this at any time and take notes as you need to. So the phosphate group is easy to draw. It has PO4, and then there are hydrogens there. R means that that's where it's attached to the larger molecules. So phosphate is easy to draw. Go for it. The ribose and the deoxyribose, those are a little different, but they're not that hard to draw. Everything is the same except for this right here. See that oxygen here? There's a hydrogen here. All hydrogens are assumed unless otherwise stated. So this is missing an oxygen here. This has an extra oxygen down there. And that is the difference between RNA and DNA, or a nucleotide and a deoxynucleotide. The nitrogen bases are here. There's 4, and we have the single ringed, which are in the bottom, and then the double ring, which are at the top. Good. Move on. So we have a nucleotide over here, which we have the pentose sugar, the phosphate group, and the nitrogen base. The deoxynucleotide, we have the phosphate group again. We have the deoxyribose here without the oxygen there, and we have a nitrogen base up there. So it might be good to draw it, considering that it's pretty much the same for both. They both have a thymine. They have everything else the same except for the nucleotide has an extra oxygen in the pentose sugar. The functions of nucleic acids. DNA contains the code for all of your proteins. That's what DNA is. It contains a protein code. The RNA helps with protein synthesis, actually helps produce the proteins. And ATP, which we'll talk about later, is our cell's energy currency. That's how we store energy. That's what we break apart when we need energy. ATP, which stands for adenosine triphosphate. And hormone reactions, when we talk about the endocrine system later on in the year, you have CAMP, which is cyclic adenosine monophosphate. So instead of having 3 phosphates like ATP, there's only 1 phosphate attached to a ribose sugar and an adenine. So that's nucleic acids. That was nice and short and sweet. So now we get to talk about proteins. We're going to spend a lot of time in proteins because they are very large and complex. And basically, we are a giant blob or giant collection of proteins and protein products. That's all we are. And that's our DNA is responsible for the production or the code for all the proteins. So the monomer of a protein, hopefully you remember, is an amino acid, from now on AA. We have a central carbon, which is our chiral carbon. Talked about that in the first screencast. Central carbon is attached to 4 different groups. We have a hydrogen group. This is our carboxylic acid group, lost the hydrogen. This is our amino group. It's a basic group, so gained a hydrogen. And this is your R group. Now, R group can be anything, and it is different in the 20 different amino acids. Everything else is the same except for the R group. Okay. Now, we put amino acids together to form a dipeptide, right? 2 amino acids together through a synthesis reaction. We can add 3 amino acids together to form a tripeptide. we can form a tetrapeptide, or we can put a lot of them together to form a polypeptide. So poly means many, which are many peptide bonds. They're put together through a synthesis reaction. And right here we have the carboxylic acid group. We have the amino group here. This is OH, this is H. OH and H forms H2O, and we form a bond between the carbon of the carboxylic acid and the nitrogen of the amino group. And that bond has a special name. Like the glycosidic bond, it is called a peptide bond. So we can form polypeptide chains by forming many different peptide bonds through a synthesis reaction. Do I want you to draw this? Oh yes, I would like you to draw it. Hit pause. Press it now. So protein shape basically determines its function. Now the triangle means change, so a change in shape will change the function of the protein. Shape is everything, and I cannot stress that enough, and how shape is everything. So the big question is, how does a protein obtain its shape? Now the protein shape is called conformation. So how does a protein get its conformation? There are 4 steps or 4 levels that have to occur for a protein to obtain its shape. The first level is called the primary level, primary being 1. It is just basically the amino acid sequence, and the amino acid sequence is determined by the DNA. So if you change the DNA sequence, if you change the nucleic acids, you will change the amino acid sequence, change the shape, and change the function. So each one of these little balls here represents an amino acid, and the little line between the little balls here represents the peptide bond. So the primary structure is just the amino acid sequence. The secondary level is a little difficult to conceptualize and visualize, but we'll try. So we have to have the amino acid chain because that's what we start off with. That is the first level. So now we have a chain of amino acid, and now we're going to start to either fold it back and forth, or we're going to start to coil it like an old-fashioned telephone cord. And as we coil or as we fold it or go back and forth, we are going to start giving it a 3-dimensional shape. How do we get these folding and coiling? But through hydrogen bonds between the amino acids of the polypeptide backbone, not between the R groups, but between the amino acids of the polypeptide backbone. So if you have a lot of small R groups, you'll have something known as a beta pleated sheet formed. If you have large R groups, and we're just talking about physical size here, you're going to have alpha helixes formed. So what does that mean? What is a beta pleated sheet and an alpha helix? So the alpha helix right here is down here, right? So we have formed a little helix like an old-time telephone cord. And this folds back and forth like this, and that is a beta pleated sheet. So we start to form these folds back and forth, and we can see how this is folding here back and forth, yeah. And this one here could coil up. So we're starting to form a shape just by the hydrogen bonds between the amino acids of the polypeptide backbone. We're not even talking about R groups yet. Not even talking about them. The tertiary level, that's the third level, so we start to fold the amino acid chain even more, and this is through R group interactions. So this is the final step to getting its shape. So what type of R groups interact? So the polar R groups will form hydrogen bonds because polar unequal sharing of electrons due to a little different electronegativities. Oh, that could be a great question. Ionic bonds are formed between acids and bases because remember, acids have a negative charge, bases have a positive charge, and the opposite charges attract. Hydrophobic interactions happen between nonpolar R groups because of van der Waals forces. You have to apply everything that we've learned so far. And disulfide bridges form between cysteines because they have sulfhydryl groups. I can show that to you tomorrow on the board. So this is a picture right here of the different types of R group interactions. We have hydrogen bonds between the polar groups. These are hydrophobic. How do you know they're hydrophobic? A lot of carbon and hydrogen. Those are nonpolar covalent bonds. They associate. This is a disulfide bridge down here. Right here, two S's together. Disulfide, 1, 2, di is 2. And then we have the ionic bond formed between the positive base and the negative acid. And you'll notice that it provides even more folds, and you add that to the already the folds from the secondary level and the coils, and you will have a protein shape. Kind of cool, actually. But I said there were 4 levels, and tertiary means 3. So what's the 4th level? Well, a protein can be made up of more than one polypeptide chain. And the polypeptide chains have to fit together. And what happens is the R groups on the different chains will react through hydrophobic interactions, through hydrogen bonds, through ionic attractions, and through disulfide bridges. An example of that would be hemoglobin. Now, hemoglobin is made up of 4 polypeptide chains. We have 2 alpha chains and 2 beta chains. And so we get something that looks like this crazy thing right here. This is hemoglobin. That's called the quaternary level, when polypeptide chains fit together. So how can we affect the shape of a protein just by a simple change in the amino acid sequence? So we're going to talk about sickle cell anemia. In sickle cell anemia, we have cells that, these are beautiful red blood cells that are nice and plump, and they can sickle under low oxygen conditions here. See them sickling there? See that real picture of sickle cell anemia cells. So we change the primary level. How? We change the DNA. If you notice here, this here is the normal hemoglobin, and we have CTC DNA, which will form glutamic acid. And here we change it to from a T to an A, and it forms valine instead. We go from an acidic to, I think this is a non-polar. So you can look that up. So we change the primary level. If we change the primary level, we're going to change the tertiary level because we're going to change R group interactions. If we change the tertiary level, we're going to affect the quaternary level because the polypeptide chains can't fit together. And that's what happens here. This is normal hemoglobin. This is sickle cell hemoglobin. Notice the shape is different because the R groups didn't interact the same way. So now we have different polypeptide chains interacting in different ways. So what happens is that instead of the red blood cells going down and being able to squeeze through the capillaries nicely, they get clogged. They're very rigid. They clog the capillaries. They can cause blindness. They can cause loss of limbs. They can cause death. All because you changed the primary level, which changed the tertiary level, which changed the quaternary level, which changed the protein shape, which changed the function. Real life example. Well, I hope that helped.